Hi, everybody. I'm Janette Doerr. I'm Senior Director of PD Upstream and Cell Line Development. Thanks for taking the time to learn about Avid Bioservices. Today, I'll be talking about accelerating biologic development and manufacturing. In today's discussion, I'll touch on several topics. First, we'll talk about Avid Bioservices and our experience supporting biologic development and manufacturing programs with our scientific expertise and end-to-end solutions and modern facilities. From there, we'll look at our early phase center of excellence and our integrated solutions for supporting programs from DNA to clinical supply. And then we'll talk about cell line development and our internal capabilities to shorten timelines. Avid Bioservices. Your CDMO partner is a strategic decision. That decision has a direct impact on the success of the program in several important ways. It influences time to clinic. Development timelines are often very compressed, especially for emerging biotech companies. Delays in development or manufacturing can push IND timelines back significantly. It influences development and scale-up risk. Decisions made during early process development can impact the program for years. If development work isn't aligned with manufacturing realities, companies can face costly rework later during scale-up or tech transfer. This decision also influences manufacturing reliability and supply. As programs move through clinical development and towards commercialization, reliable and consistent manufacturing becomes critical. The Avid advantage. Avid combines modern biologics infrastructure, scientific expertise, platform alignment, and operational excellence to accelerate biologic development and manufacturing while reducing program risk. Avid is a US-based CDMO. We have integrated cell line development and formulation development for end-to-end solutions to accelerate timelines. Our deep expertise and solid regulatory track record reduce program risk. Our facilities and equipment are aligned across sites and from early stage to manufacturing to support program transition from IND to commercial manufacturing. Avid supports biologic programs across the key stages of development and manufacturing, helping clients progress efficiently from early development through commercial supply. We take a phase approach to biologic programs. DNA to IND, development and IND readiness. This phase focuses on development activities that prepare a program to enter the clinic. That includes work such as cell line development, process development, formulation development, and analytical method development. IND to commercial, clinical manufacturing and scale-up. Once a program enters clinical development, the focus shifts toward manufacturing clinical trial material and continuing to optimize and scale the manufacturing process as the program advances through clinical phases. And the last phase is product to patient, commercial, manufacturing, and supply. This focuses on commercial manufacturing and reliable long-term supply once therapies reach the market. Avid is a US-based CDMO with operations in Southern California with infrastructure that can support early phase speed and long-term manufacturing. We operate across 2 sites that are designed to complement each other, enabling both agility and scale. Our facilities are designed specifically for biologics. We support multiple stages of drug development, including cell line development, process development, clinical manufacturing, and commercial supply. We have integrated development and manufacturing infrastructure, which is designed to support program progression, and it enables scaling from early development through commercialization. As your program advances through clinical stages, manufacturing requirements change, and our infrastructure is designed to scale with that progression. Avid's manufacturing infrastructure includes multiple production lines and reactor scales designed to support clinical supply, scale-up, and commercial manufacturing. We have multiple manufacturing lines and reactor scales. We have flexibility to support different program sizes. We have the ability to scale processes as programs advance, and our manufacturing environments are designed for biologics. Avid has extensive experience supporting biologic development and manufacturing programs, with the infrastructure, teams, and operational scale required to support biologics. Avid has produced over 600 batches. We have over 30 years of experience in manufacturing biologics. We have 23 years of successful inspection history. We have 20 years of commercial manufacturing. We make 7 commercial products, and over the last 10 years, we've had 043s during regulatory inspections. Quality is fundamental to Avid's operations, with strong regulatory compliance and systems designed to support clinical and commercial biologics manufacturing. Quality culture is embedded across the organization. We have robust quality management systems. We're regulatory compliant and inspection ready. We have experienced quality and regulatory teams. And we have systems designed to support clinical and commercial programs. And this is a timeline of our last inspections, with specifically, we had 2 inspections in 2025 by FDA and the CDPH. Avid scientific teams bring experience across multiple biologic modalities, allowing them to support diverse development programs. We have experience making monoclonal antibodies, ADCs, multiple specifics like bi and trispecifics, Fc fusion proteins, recombinant proteins, different isotypes of IgDs, IgMs, minibodies, enzymes, and cytokines. On to our early phase Center of Excellence in Costa Mesa. The Early Phase Center of Excellence was designed to accelerate early-stage biologic development. It has integrated development capabilities in one environment. We have modern equipment and infrastructure, and we support DNA to IND development activities here. We enable efficient collaboration across scientific teams, and we support scale-up to our Myford late-stage facility. And we just launched this earlier in 2026. This is a top-down view of our Center of Excellence facility. We added cell line development and formulation development to our offerings, giving clients end-to-end partner from molecule to market. What I want to highlight here is the 25,000 square foot expansion we plan to bring online in 2027, where we'll consolidate all of our development activities under one roof. We're expanding our capabilities and capacity to serve clients easier, faster, and even more seamlessly. Avid's new integrated model brings every discipline from cell line development, process development, formulation development, and manufacturing together to ensure quality and scalability from the start. We've added cell line development to our offerings, giving clients a true end-to-end partner from molecule to market. Our CLD system is based on the leap-in transposase technology, and we use the advanced instrument suite of cell line development equipment, including the VITS, metric, and the ICON. And we'll talk more in detail about cell line development in a bit. We've added formulation development to our offerings, again, for a true end-to-end partner from molecule to market. For a standard antibody, we've generated a handful of platform formulations with different buffer components, stabilizers, surfactants, and viscosity, where we'll test molecule stability in real time and accelerated studies. We've invested in the standard analytical equipment to support both cell line development and formulation development activities. And here's a list of the equipment and the assays that we can support, but I won't go over them in detail here. Avid aligns development platforms and manufacturing equipment so processes developed at our Center of Excellence can transfer efficiently to our manufacturing environments at Costa. Our equipment is aligned across the facilities. Our process development is designed with manufacturing in mind. We minimize tech transfer challenges, and we reduce development rework during scale-up. And all of this enables faster progression from development to manufacturing. So shown here in our PD lab, our largest scale is a 100-liter bioreactor, which matches the same equipment as the smallest scale in manufacturing. And that's also true for downstream. Avid's goal is to go from DNA to IND readiness in approximately 40 weeks. Shown here is an integrated timeline of major activities of cell line development to IND readiness. We strive to go from DNA to completion of GMP in just 33 weeks. Avid's integrated development capabilities and coordinated teams support an efficient progression from DNA to IND readiness, helping clients reach clinical trials faster while minimizing risk. Now we'll talk a little bit more in depth about cell line development. As I mentioned before, our cell line development platform is based on the leap-in transposase technology. This technology allows for multiple gene copies to be inserted in each cell, leading to robust titers. It allows for accelerated workflows since the stable clones resemble the transfected pools, so PD activities can begin in parallel with cell line development activities. And the leap-in transposase technology facilitates expression of complex molecules with multiple chains. This is a high-level block diagram of our process flow for cell line development of a standard antibody, where what I want to highlight is one thing where we can do the fed batch pool production and single-cell cloning in parallel to accelerate timelines. We also accelerate timelines by performing stability testing, starting with the top 24 clones and then whittling it down from there. So we're saving time in multiple spaces in the CLD workflow. Using our standard MAB and accelerated cell line development workflow, we can generate stable pools along with fed batch analysis in as little as 5 weeks, and single-cell clones in RCB in 14 weeks, and we can complete stability studies in up to 20 weeks. This slide shows a little bit more detail in our stable pool generation. For a standard MAB, we'll construct 2 expression vectors with varying genetic elements. We'll use the neon electroporator to transfect CHO-K1 GS cells with our gene of interest, along with a transposase mRNA, and we will generate 2 stable pools per molecule. Once the cells have recovered from selection, they are expanded to 14-day fed batch production runs. And then we assess the pool for titer on days 7, 10, 12, and 14 by either the ICON or the octet. We'll then perform a single-step purification so we can look at some product quality using CE-SDS or SDS page, SEC. We can even go further and look at N-glycans, charge variants, or for bispecifics, heterodimer content. Off to single-cell cloning. Our single-cell clones are generated and analyzed by advanced instrument platform CLD equipment. We'll use the VITs to deposit cells into plates and generate single-cell plate maps. We'll use the metric to confirm monoclonality by imaging on days 0, 1, 2, 3, and 7. Clones are then expanded for further analysis and banked for risk mitigation. This leads us to our first clone ranking step. So we'll take 100 clones and analyze them in 7-day fed batch cultures in deep well 24 plates. Clone assessment will then include titer testing on days 5 and 7 by the octet or the icon. We'll measure growth and viability. We'll calculate IVCD and specific productivity. Again, we'll confirm the monoclonality of the top 24 clones that have been selected by titer. And then we'll cryopreserve 4 vials of the top 24 clones. And we can also start to look at product quality here too by doing our single-step purification and then running the analytics that I mentioned previously. Next, we take the top 24 clones and take them into our second clone ranking step. So here, clones are analyzed in a 14-day fed batch culture in spin tubes. The clones are analyzed by titer on days 7, 10, 12, and 14. Then we'll do some single-step purification and look at product quality again. We'll calculate the IVCD and specific productivity, and then we'll rank the top 5 and in some cases the top 10, and those will move on to RCB banking. Clone stability testing. Another way that we shorten timelines is to initiate the clone stability testing on the top 24 clones. And then once the top 5 clones are selected, we'll continue on with the stability testing of those top 5 clones. We'll passage the clones in media with and without glutamine up to 60 population doublings in deep well plates. And then the analysis includes looking at consistent growth rate from population 0 to 60, consistent productivity between 0 and 60 populations with and without glutamine in a 7-day fed batch deep well plate. For more complex molecules, again, we can look at product quality by doing a single-step purification. We'll also look at consistent copy number between population 0 and 60 with and without glutamine by ddPCR. We'll do some cDNA sequence verification on that population 60. And then we can also do TLA analysis by outsourcing to Sargentis. RCB generation and release. So RCB generation is initiated on the top clones, and the top 5 will move on to RCB generation. So we'll bank down 30 vials for each clone, and then release testing includes identity by cDNA sequencing using Oxford nanopore sequencing, sterility and mycoplasma testing, and then we'll look at growth and viability after thaw and passage them twice. This is a detailed timeline of our accelerated standard MAM CLD process. Stable pools can be generated and evaluated in approximately 5 weeks. Single-cell cloning is initiated along with pool fed batch run to expedite timelines. And RCBs are generated by 14 weeks, and clone stability studies are completed by 18 to 20 weeks. Thank you, everybody, for listening. If you have any further questions, please contact us on the Avid Bio website.